Hello students, today we'll start with a new chapter, chapter 3 of your NCERT, that is human reproduction. Children, as you all know, humans are sexually reproducing organisms and they show sexual dimorphism. Sexual dimorphism means male and female are morphologically different. Humans belong to the kingdom Animalia. Phylum is Chordata, class Mammalia because they give birth to young ones. Order is Primates because menstrual cycle runs in the female of the primates and they have 5 digits. Family is Hominidae. Hominidae, that is they are tailless. They do not have tail. Genus is Homo and species is Sapiens. So humans are viviparous animals. They are viviparous. They give birth to young ones. They are sexually dimorphic, male and female are externally as well as internally distinguishable. Dioecious means male and female organs are present in separate body. Now, what are the reproductive events in human beings? So, reproductive event starts from gametogenesis and gametogenesis starts at puberty, at the adolescence age. In males, spermatogenesis takes place, that is the formation of sperms, and in females, oogenesis takes place, that is formation of egg. Next is insemination. Insemination is transfer of sperms into the female genital tract. Fertilization, fusion of male and female gametes. Implantation, formation and development of blastocyst, and this blastocyst is attached to the uterine wall. This is called implantation. Gestation period is of 9 months, approximately 9 months, and it is known as embryonic development as well. Parturition, delivery of the baby or birth of the baby. So all these events are included in reproductive events in humans. Gametogenesis, insemination, fertilization, implantation, gestation and parturition. Sperm formation continues in old men, but formation of ovum ceases in women after menopause. That is, when menstrual cycle stops, the formation of egg stops in the human female. This is approximately at the age of 50 years. So first we'll start with male reproductive system. In male reproductive system, a pair of testes are present, accessory ducts, glands and external genitalia. All these are the components of male reproductive system. A pair of testes, accessory ducts, glands and external genitalia. Testes. These are the male reproductive organs. Testes. They help in sperm formation. There are 2 functions of testes. First, spermatogenesis, that is formation of sperm, and second, secretion of androgens. Androgens are male sex hormones. So testes form sperms plus they secrete male sex hormones, that is androgen. The main male sex hormone is testosterone. Main or principal male sex hormone is testosterone. So testes are located in the scrotum, which is an extra-abdominal pouch. Testes are present in human males in the extra-abdominal pouch, that is scrotum. But testes are formed inside the abdomen during the fetal stage. Testes are formed in the abdomen during the fetal stage, but they descend into the scrotum during the 7th month of fetal life. So this indicates the abdominal origin of the testes. Temperature is maintained 2 to 2.5 degree lower than the body temperature in scrotum. In scrotum, the temperature is 2 to 2.5 degree lower than the body temperature, and sperm formation requires the temperature less than the body temperature. Therefore, testes are located in the scrotum. Low temperature is required for sperm formation. There are some mammals which have intra-abdominal testes. There are some mammals which have their testes in the abdomen. Example, elephants, whales, egg-laying mammals. In humans, the development of testes takes place in the abdomen. But in the 7th month of fetal life, testes descend in the scrotum. So, testes are found in the abdomen, but in the 7th month of the fetal life, they descend into the scrotum. Why they descend into the scrotum? Because for the formation of sperms, temperature required is less than the body temperature. And in scrotum, the temperature is 2 to 2.5 degree less than the body temperature. The hormone testosterone helps in the descent of testes through the inguinal canal. Children, what is inguinal canal? I told you that testes descend from the abdomen into the scrotal sac. So they come from abdomen to the scrotal sac through inguinal canal. This descent of the testes is regulated by the hormone testosterone. Means testosterone helps in the descent of the testes from abdomen to the scrotum through inguinal canal. So inguinal canal is narrow duct joining scrotum to the abdomen. Inguinal canal is the narrow duct joining scrotum to the abdomen. Once the testes descend, then the tissue fills the inguinal canal. Fibrous tissue fills the inguinal canal. Once the testes descend into the scrotum, then this inguinal canal is filled with the fibrous tissue. But through this canal, vas deferens, blood vessels, nerves can pass. This canal is major part of this canal is filled with fibrous tissue, and small part is open to run vas deferens, blood vessels, nerves. Vas deferens brings the sperm from the testis to the male urethra. Blood vessels provide nutrition to the testis, and nerves which can regulate the functioning of the testis. So once the testis descend from the fibrous then the fibrous tissue fills the inguinal canal, which is called closure of inguinal canal. But through this inguinal canal, vas deferens, blood vessel, nerves can pass. And these three things together form the spermatic cord. So spermatic cord is fibrous cord in the inguinal canal which contains vas deferens, blood vessels and nerves. Blood vessels include both vein and artery. Through this vas deferens, blood vessels, lymph vessels, nerves and cremaster muscle passes. See, around the scrotum there is a muscle, dartos muscle. Dartos muscle is sensitive to temperature. This is sensitive to temperature. When the temperature decreases, this dartos muscle contracts and brings the scrotum near to the body wall. It brings scrotum near to the body so that scrotum can absorb the heat from the body. But when the temperature rises, this dartos muscle relaxes and moves scrotum away from the body so that temperature does not rise in the scrotum. Muscle around the scrotum is dartos muscle. During low temperature, dartos muscle and cremaster muscle contract and brings the scrotum near to the body so the temperature is increased. During high temperature, these muscles are relaxed. In this way, regulation of temperature of testis takes place because sperm formation is highly sensitive to temperature. Cryptorchidism. What is cryptorchidism? Failure of the testis to descend into the scrotal sac is known as cryptorchidism. These males are sterile. Because the testes do not descend into the scrotum and they are unable to form the sperms. Failure of the testes to descend. Therefore, sperm formation will not take place due to high temperature in the abdomen. This disease is known as cryptorchidism. Next is monarchidism. What is monarchidism? Presence of single testis is known as monarchidism. Natural monarchidism is found in ascaris. In ascaris, only one testis is present naturally. Function of testis. I have told you there are two functions of testis. First, the formation of male gametes. Therefore, male gametes, that is sperm. Therefore, testes are called primary sex organs. Second, secretion of sex hormone, that is androgens. Most important androgen is testosterone. Now children, this is very important This is the diagram of testis, LS of testis, and this can come in board exam under 3 marks or 5 marks with explanation. So this is, in this diagram I have shown the longitudinal section of the testis. See, the outer covering of the testis is known as tunica albuginea. It is known as tunica albuginea. Tunica albuginea innervates, it moves inside or innervates the testis to divide the testis into testicular lobules. See, these are testicular lobules. Tunica albuginea innervates to divide testes into testicular lobules. There are at least 250 to 300 lobules, testicular lobules in each testis. Now this testicular lobule has seminiferous tubule, coiled tube-like structure called seminiferous tubule in each testicular lobule. There are 1 to 3 seminiferous tubules in each testicular lobule. How many seminiferous tubule? 1 to 3. Total how many testicular lobule? 250 to 300. These seminiferous tubules are highly coiled and they are made up of germinal epithelium. They have germinal epithelium which will give rise to sperms. They are lined by germinal epithelium which will give rise to sperms. This seminiferous tubule comes out of the testicular lobule in the form of tubule recti. Tubule recti. This is a straight tube. Seminiferous tubule was coiled, but this recti is straight. These recti meet together and form retitestis, network-like structure called retitestis. Retitestis leads into vasa efferentia. They leads into vasa efferentia. These are also small tubes which arises from retitestis. These vasa efferentia then meet into or form, they merge into epididymis. They merge into epididymis. Epididymis has 3 portions: caput, corpus and cauda. Caput, corpus and cauda epididymis. This epididymis then ascends into the abdomen. It ascends into the abdomen and forms vas deferens. It forms vas deferens. So this is the structure of testis. Outermost covering is tunica albuginea. Tunica albuginea innervates to divide the testes into testicular lobules. These testicular lobule, each testicular lobule has 1 to 3 seminiferous tubules. These seminiferous tubules are coiled. They emerge out of the testicular lobule in the form of straight tubes called tubulae recti. Tubulae recti form retitestis. From retitestis emerge vasa efferentia. Vasa efferentia lead to epididymis, and epididymis leads into vas deferens. Vas deferens ascends into the abdomen. It ascends, it runs upwards into the abdomen. Now, testis is surrounded by a connective tissue sheath called tunica albuginea. This tunica albuginea divides testes internally into testicular lobules. Each testicular lobule is also surrounded by tunica albuginea. So, it innervates into the testis. Therefore, each testicular lobule is surrounded by tunica albuginea. Testicular lobule may be 250 to 300 in number. Within each lobule lie highly coiled seminiferous tubule. They are 1 to 3 in number. Seminiferous tubule come out of the testicular lobule to form narrow straight duct called tubular recti. This I have told you, shown you that seminiferous tubules are highly coiled. When they emerge out from the testicular lobule, they emerge out in the form of a straight tube called tubular recti. Tubular recti continue to form a network of ducts called retitestis. From retitestis arise small narrow ducts called vasa efferentia. Vasa efferentia leads into a highly coiled duct present in epididymis. This epididymis has 3 parts: caput, corpus and cauda. Vas deferens or sperm duct comes out of the epididymis. Vas deferens enter or ascends into the abdomen. It ascends into the abdomen. Sperm formation takes place in seminiferous tubule. Seminiferous tubule has germinal epithelium. Germinal epithelium is cuboidal epithelium which forms the gametes. Release of sperm from seminiferous tubule is called spermiation. Release of sperm from seminiferous tubule is called spermiation. So in this I have shown the TS of seminiferous tubule. Seminiferous tubule is lined by germinal epithelium. This is germinal epithelium, lowest layer, round cells. These are germinal epithelium. It is made up of cuboidal epithelium. This will divide and give rise to sperms. In between germinal epithelium, there are pyramidal shaped cells which are known as Sertoli cells. These are known as Sertoli cells. These Sertoli cells function as nurse cells. They function as nurse cells, nurse mother cells. When the sperms are formed, they embed their head into the Sertoli cell to gain nutrition. When sperms will be formed in the seminiferous tubule, they will embed their head into the Sertoli cell to gain nutrition. So this is the structure of seminiferous tubule, and these cells I have shown different stages of sperm development, which we'll discuss later on. Between seminiferous tubules lie Leydig cells or interstitial cells. Leydig cells or interstitial cells are endocrine in nature. They secrete hormones. So between seminiferous tubule, Leydig or interstitial cells are present. In seminiferous tubules, two types of cells are present. Sertoli cell, pyramidal in shape. On the lining of seminiferous tubule is present germinal epithelium. Germinal epithelium divide meiotically, and various stages of division of these cells are seen in between the Sertoli cells. These stages we will discuss later on. Sperm embed their head in Sertoli cell to gain nutrition. Some sperms are present in the lumen of seminiferous tubule. Leydig cells are endocrine cells. They secrete androgens. They secrete male sex hormone, and main androgen, as you all know, is testosterone. Testosterone is a steroid hormone. It is necessary for spermatogenesis. It is also required for development of male secondary sexual characters. Testosterone is also required for development of male secondary sexual characters like beard, moustaches, deepening of voice, etc. Sertoli cells are supporting cells. These nourish the developing sperms. They also secrete inhibin hormone, which inhibits follicle stimulating hormone from the pituitary gland. They also secrete Sertoli cells. They also secrete ABP, androgen binding protein. It binds with testosterone and keeps the level of testosterone high in the seminiferous tubule, which can help in the development of the sperms, which is necessary for spermatogenesis to occur. So Sertoli cells, they help in the nourishment of the sperms. They secrete inhibin hormone, which inhibits the secretion of FSH. They also secrete ABP, androgen binding protein, which binds to the testosterone and maintains the concentration of testosterone in the testes high, so that it can help in spermatogenesis. During embryonic stage, Sertoli cells also secrete. This is very important from the competition point of view, but this is not given in your NCERT. During embryonic stage, Sertoli cells also secrete Mullerian inhibiting factor, MIF. Mullerian inhibiting factor. See what happens during fetal life. Primordial cells of both, precursor cells of both male and female reproductive ducts are present. But if the fetus is male, then Sertoli cells will form Mullerian inhibiting factor, which supports the formation of Mullerian duct system, that is which suppress the formation of Mullerian duct system, that is female reproductive ducts. Children, this is very important. What I have told you is that during embryonic life, Sertoli cells secrete Mullerian inhibiting factor. What is this Mullerian inhibiting factor? During fetal life, both the cells, precursor cells of female reproductive system, duct system, and male reproductive duct system are present, which can form female reproductive duct and male reproductive duct. But if the fetus is male, then Sertoli cells will secrete MIF, which will suppress the formation of female reproductive ducts. So MIF suppress the formation of female reproductive ducts in case of the male fetus. Now, this is the flow chart showing the path of the sperms. Sperms are formed in seminiferous tubule, then they come into tubuli recti, then retitestis, then vasa efferentia, then epididymis and vas deferens. From seminiferous tubule to vasa efferentia, this is intratesticular duct system, means all these 4 ducts are present inside the testis. Epididymis, which is lined by pseudostratified epithelium. Epididymis is lined by pseudostratified epithelium, which you have studied in +1. It is single layered, simple epithelium, but some cells are short in size and some are big in size. So it looks like double layered, but it is single layered. This pseudostratified epithelium has stereocilia. Epididymis secrete factors for functional maturity of sperms. So some maturation of sperm also occurs in epididymis. It secretes some factors which help in the maturation of sperms. Vas deferens is also lined by pseudostratified epithelium with stereocilia. Outer to this epithelium is a layer of smooth muscle. So pseudostratified epithelium, outside pseudostratified epithelium in vas deferens there are smooth muscles, and contraction of these smooth muscles propels the movement of the sperm. Vas deferens re-enters abdomen, loops over urinary bladder, forming a bulbous part ampulla. This I will show you in the diagram. See, this is testis, this is scrotal sac, and this is vas deferens. This vas deferens ascends into the abdomen and loops over the ureter. This loops over the ureter and swells to form the ampulla. This ampulla opens into the ejaculatory duct. This ampulla opens into the ejaculatory duct, in which the duct of seminal vesicle also merges. See, this is vas deferens, this is ampulla. Ampulla opens into ejaculatory duct, and in ejaculatory duct merges the duct of seminal vesicle. So ampulla open into ejaculatory duct, in which duct of seminal vesicle also opens. From the prostate gland, see, this is the prostate gland. Seminal vesicles are paired glands, whereas prostate gland is single gland. From the prostate glands, many ducts open into the ejaculatory duct. Now this is the ejaculatory duct and many ducts. See, I have shown by drawing the lines here. Many ducts from the prostate glands open into the ejaculatory duct. In ejaculatory duct, Copper's gland also pours its secretion. In ejaculatory duct, Copper's gland also pours its secretion. This Copper's gland is also known as bulbourethral gland. Now this comes into the urethra and it opens out through the penis. So now the ejaculatory duct becomes a urethra which opens out through the penis. See, here I have also shown. This is the testis, this is the vas deferens. Vas deferens forms the ejaculatory duct. Seminal vesicle also pours its secretion into the ejaculatory duct. Then prostate glands also pour their secretion into the ejaculatory duct, and it becomes urethra and opens out through urethral meatus. Now children, reproductive gland we will discuss in the next video. Male reproductive glands we will discuss in the next video.